Welcome back. In this particular video, we're going to start looking at the results from the metagenomic read mapping jobs. This will be in two parts, looking at the report and then the other things that are part of the job results. So let's go find our job. We could start up here in workspaces and navigate to wherever the job is. You could go to My Jobs here, or you could click down here on the Jobs monitor. So you can see I've run a lot of jobs. If I ran this a while ago, it might be hard for me to find it. I could use the filter name up here, but I may have forgotten exactly what I named it, especially if it's at a time when I'm starting to work on a publication. So I can click on this down arrow here, and that shows all the different services available in BV-BRC. And I can look at narrowing it down to metagenomic read mapping. So I click on this, and it's going to show me all the jobs I've launched to that particular service. And this is the first one we want to look at when we did this read file against the card database. So I'm going to click on that and then click on View. In the later videos, we'll talk more about what's happening up here. But for this video, we want to get right to the Metagenomic Read Mapping Report, the HTML file. I could get to it here or here by clicking on the view icon. Now, this is what it looks like, and let me move this over so you can see this. It's telling me this is a hyperlink to the data set that I use to generate it, the read files, and here is the table. So let's discuss a little bit about what these things mean. It's important that you understand the column heads. We're going to be switching back and forth between tabs, so keep up. This is the tutorial for the Metagenomic Read Mapping Service. And I'm going to scroll down until we get to the description of these things. So here's the report here, and here's what it looks like. And this is the KMA standard sample report format. Remember that KMA is what is used in this service, and it's a way to distill information. So it's faster than BLAST because it's distilling the information across the genes to a smaller data set. So in the first column is the template column. And that is the identifier of the template. You've got to get the template, which is the reference data, versus the query, which is what your reads are mapping to. So let's look at this. This is the template here. And you can see that the first thing it says is card. And that's indicating that it's the card database. And there's often an accession number after that. And if I were to click on that, it would open up a table that showed me all the genes in the database that had BLAT results to that particular template across all the antibiotic resistant genes. So I'm going to go back on this. The second column is the function, and that is the name. It's not the name necessarily that BV-BRC provided, but in this case, it's the name that card provided. So let's look at that. So this is the name assigned by that. Okay. The third is the genome that contains the template gene. BV-BRC is all about genomes. So we can't just have a gene hanging out there all by its own in the database like you'd find at GenBank. Here it has to be part of a genome. So this is the genome that had the gene. And when we look at this, we could go in and click on that, and that's going to rewrite the page and show us that genome. Oh, look at this. This is an interesting thing. It showed us not one genome, but many genomes because it was looking for Streptococcus pneumonia R6. And this is something all biologists need to understand, that these names are not unique identifiers. Let me go over here and click on the plus icon. and add genome ID and then click it off, these are unique identifiers for each of these genomes. And each of these have an R6 in it. So it was finding a hard time recognizing that. So it gives us each of those. So let's go back here. And then let's go back to the description of those things. So the fourth column is a score. And this is a global alignment score. And it's a conclave score. This is described in the KMA paper, but it's looking at the accumulated alignment score from all the reads that map to that particular template. In this case, that template is a particular gene. And if we wanted to look at that, here are the scores. Then we have expected. Expected is the alignment score if all the reads were normally distributed over the entire database. So that's what you would expect. And so you're looking at the score and then the expected score. So in some cases, it's a lot more than you would expect. And in others, it's pretty close to what you would expect. Then we have the template length, and that's the template gene length in nucleotides. So you can see here are the template lengths. So this is the nucleotide for this particular sequence. And this is the nucleotide length for this particular PBP2B sequence. The template identity. Okay, the template identity is the number of bases in a consensus sequence that are identical to the template sequence divided by the template length. So it's trying to give you a sense of how many nucleotides are matching. So you can see here that it's like 86%, 99%, 96%. So some of these are pretty good. All of them appear to be pretty good. Template coverage. Those are the percentage of bases in the template that is covered by the consensus sequence. So if it's above 100%, this is indicating that there are more insertions than deletions. So let's look at this. So the template coverage, do we have any here that are above 100%? I sometimes see that, but we don't see any of that here. And then we have query identity. Now we've switched from talking about the reference gene and to what the reads are covering. So this is the percent identity between the query and template sequence over the length of the matching query sequence. Okay, it's the number of bases in the template sequence that are identical to the consensus sequence by the length of the consensus. So let's look at that. So here's the query identity. Okay, and now we need the query coverage. And the query coverage is the length of the matching query sequences divided by the template length. And the reason that I like both this, the query coverage and the template coverage, is the score gives you an indication of if you might have insertions or deletions. So if it's above 100%, it means that there are more deletions than insertions in the query sequence. So let's look at that. And some of them have a higher than 100% coverage. Okay, so that might mean you might want to do an MSA or look more deeply into those particular genes. And then you have the depth, the number of the times the template has been covered by the query. So here's the depth. And this is something that you see a lot in assembly reports. They always want to know how deep the coverage is. And you can see that some of them have a great depth. And some of them, like this RLMA, have a kind of shallow depth. Not a lot of reads mapped to this thing. And then finally, We have the Q value where it's telling you it's from this McNemar's test to see if the template is a significant hit, and then the P value for that. So you have Q values and P values. All right, so now we understand what each of these things mean. And if you scroll to the bottom, you can also see that some of that information is here as well. And we've shown you that clicking on these things are hyperlinks to other pages. But another thing I wanted to show you was that you have a number of different genera here that these things are mapping to. It's looking across everything. It doesn't know exactly what you want. So it's looking across everything in card and trying to show you things that there are significant hits against. And we can see that there's some Haemophilus, some Streptococcus, some Neisseria. But if you look at this, there are a lot of PBP genes, PBP3, PBP2X, PBP2. Is this an indication that there are a lot of penicillin binding proteins in this particular genome? Not necessarily, because each of these could be against the same gene. Just because it's bringing in everything, stuff from Haemophilus, stuff from Streptococcus, stuff from Neisseria, if there is a lot of homology between the PBP genes in those genomes and in our read set, we'll see multiple things of that. So it's important, especially if you're going to publish a paper on this, to do some due diligence and look at what things these are actually hitting to, if that's of interest to you. Okay, so this is what the report looks like. And this is what it looks like when we're looking at a job that we used our read sets against card. Would look the same if it was VFDB. Let's go look at jobs again. So this time I'm going to go to my jobs here. I'm going to filter on metagenomic read mapping again. And this time I want to look at the genome report when I ran it against a feature group. And here I named it here. So I'm going to click on this and then click on the view icon. And this time I'm going to load the report by clicking up here. And so looks the same, right? But a lot fewer results. Now, I want you to remember that when we did the feature group, there were a lot of genes in there. And we aren't seeing those same genes here. We don't see any of the tetracycline genes. There are a number of things that we are not seeing in this that we asked it to look for. But these are the ones we're seeing. So it's down to 1, 2, 3, 4, 5 of those. And here are the scores and the expected, and these are all from Haemophilus influenzae. Now, this is a gene ID within BV-BRC. So I should be able to click on this, and it'll rewrite the page to show me that particular gene. And here is that gene here. So it's giving me the information about that gene. And note that when you're at the feature page for any gene that is part of a genome in BV-BRC, you have a genome browser, you have the compare region view that's looking at the gene neighborhood. We won't see any transcriptomics or interactions, but we might see some domains. Let me look at the compare region viewer, because this one's kind of fun. It takes the central gene and then it's blasting it on the fly against all the genomes in the database, all the reference and representative genomes, and it's showing you the upstream and the downstream regions. We have tutorials on this and videos on this that I encourage you to look at. And sometimes you'll see domains and motifs. With private genomes, sometimes you don't see them. But this is pretty valuable when it pops up. Well, let's go back to that particular job and the feature group here. view. And look at that again. That's it really for this particular job. So I want to point out that when we can provide links, if it's within the feature group that was generated in BV-BRC, those will all have unique identifiers. So we will link to the gene and the genome. It's different when it's the FASTA file. So let's go back to the jobs. And I ran one here with a FASTA file, which are different genes. And this, well, they're the same genes, but from a different source. And this is a way that you can compare data outside of BV-BRC. Let's say you have data from UniProt or GenBank or some publication that you want to compare against them. This is a way that you can do it. And the mapping report will look a little bit different. Notice that here it's just taking what I gave it as a sequence identifier. My sequence identifier didn't have a name and it couldn't link to a genome in BV-BRC. So this part will look different. But look again, it's the same genes that we were seeing in the feature group. So this is what it looks like when you do a FASTA file and look against the reads. So in the next video, we'll talk about the other files that come when you complete the metagenomic read mapping job. Thanks for joining. Bye.